Welcome student for today's lecture. Student, today we are going to see the next point of our topic, that is the genetic code. Actually आपण प्रोटीन सिंथेसिस पाहत होतो, बरोबर आहे. आणि प्रोटीन सिंथेसिस मध्ये आपण पार्ट फर्स्ट, पार्ट सेकंड आणि पार्ट थर्ड पर्यंत आपण ट्रान्सक्रिप्शन प्रोसेस पाहिली. आता प्रोटीन सिंथेसिसचा राहिलेला पार्ट हा पुढे आपण अजून ट्रान्सलेशन प्रोसेस बघणार आहोत. पण त्याच्या अगोदर जेनेटिक कोड हा कन्सेप्ट तुम्हाला समजणं गरजेचं आहे. त्यामुळे इथं आपल्या सिलेबसमध्ये जेनेटिक कोड हे दिलेले आहे. ठीक आहे. तर आता आपण सुरुवातीला कॅरेक्टरिस्टिक्स ऑफ द जेनेटिक कोड, ऑफ द प्रॉपर्टीज ऑफ द जेनेटिक कोड. सो व्हॉट आर द कॅरेक्टरिस्टिक्स ऑफ द जेनेटिक कोड? So genetic code means what? That is, the genetic code is are mainly in the form of the triplet, and they are the always present on the mRNA. म्हणजे जे genetic codes जे आहेत, this genetic code, where they are present, they are present on mRNA. They are present in the mRNA. So if we read, that is the genetic code. These are read that is from the 5 prime end to the 3 prime end. So it is the genetic code which are present.
Piece of genetic code. 1. The code is triplet. 61 codons code for amino acids and 3 codons do not code for any amino acids. Hence, they function as top codons. Example: UUU, phenylalanine, GGG, glycine. 2. The code is degenerate. In triplet code, 64 codons are possible. Thus, each amino acid is coded by more than one codon. Example: serine is coded by UCU, UCC, UCA, UCG, AGU, and AGC. 3. The code is non-overlapping. The code should be always read in groups of 3 nucleotides from a fixed starting point. The 3 letters of the codon are considered as a unit and should be read as if they are exposed through a reading frame. 4. The code is commaless. In between triplet codes, no letters are wasted. No punctuations are needed between any 2 codons. 5. The code is non-ambiguous. One codon codes for only one amino acids. Hence, it is specific and non-ambiguous. 6. The code is universal. The same code applies in all kinds of living organisms. Example: UUU codes for phenylalanine in all living organisms. 7. Initiation codons. The codons which initiate the process of polypeptide chain formation are referred as starting or initiation codons. AUG is the starting codon and is present as triplet codon on all mRNA. AUG codes for methionine, MET. 8. Termination codons. The triplet code, which is at the end of mRNA and does not specify for any amino acid and serves for termination of polypeptide synthesis, is the termination codon. The codons UAA, UAG, and UGA are termination codons. UAA is called ochre, UAG amber, and UGA as opal.
In RNA, that is thymine is absent. Okay, so see that is here. The mRNA, if we see RNA, that is in RNA there are the 3 types: mRNA, rRNA, and tRNA. Okay, so here on the mRNA, that is here, there are the 4 nucleotides, that is the adenine nucleotide, guanine nucleotide, cytosine, and uracil. That is thymine is absent. Thymine is absent in the RNA. So here if we read, that is in the DNA, if we see in the DNA, that is in the DNA purines, these are the adenine and guanine, and pyrimids, these are the thymine and cytosine. Okay, so here in the DNA, that is genetic code, these are read, that is based on these 4 nucleotides, adenine, guanine, thymine and cytosine. While in RNA, that is adenine, guanine, cytosine and uracil is there. Uracil is there. That is here in the DNA, thymine is present. In RNA, thymine is replaced by the uracil. Okay, students. So see, that is the characteristics of the genetic code. So genetic code, one by one आपण एक characteristics बघूया. So first characteristic, that is the genetic code are in triplet. Triplet, that is triplet means what? In every living organism, that is genetic codes are always triplet. So if we see, that is the mRNA. So see here the mRNA. Okay. So in mRNA, here the first 5' end, here is the 3' end. So genetic code, if we see, that is a read, that is from the 5' end to the 3' end, and the 3 successive nitrogen bases which form the one codon. 3 successive nitrogen bases, that is, for example, at the 5' end, here is the nitrogen base, that is the AUG. So here AUG, these are the 3 successive nitrogen base, and these 3 successive nitrogen base which produce the one codon. That is here, these 3 successive nitrogen base, it is known as the codon. And let's come into upon codon. So here the codon, these are always triplet form, that is 3 successive nucleotides. म्हणजे 3 successive nucleotide मिळून एक codon तयार होतो. So the codon, these are, that is the first characteristic, that is the codons are in triplet form. That is the codons, that is genetic code is always triplet. पहिला characteristic काय येईल, genetic code is always a triplet. And this genetic code, these are read, that is from the 5' end to the 3' end. Okay. Then next see, that is here. आता आपण हे जर बघितलं तर हा एक जेनेटिक कोड झाला. याच्या पुढे दुसरा येईल, फॉर एक्झाम्पल CGU. हा दुसरा असेल जेनेटिक कोड. It is also in the triplet form. Okay, so each codon, this codon is in the triplet form. This codon is the triplet form. That is 3 successive nitrogen bases which forms the codon. So the genetic code is always triplet. Then next, that is here. Second characteristic, that is about the polarity. That is genetic code having polarity. Second characteristic, genetic code having polarity. Genetic code having polarity. मग genetic code मध्ये polarity काय असते? If we see, that is the genetic code, these are always in direction, that is from 5' end to the 3' end. म्हणजे हा 5' end झाला mRNA चा, it is the 3' end. Okay, so these are read from the 5' end to the 3' end. Otherwise the message will be changed. जर समजा एखादा जर genetic code change झाला, म्हणजे for example AUG, it is the initiation codon. AUG. Okay, it is the initiation codon. So AUG, it codes for amino acid. AUG codes for amino acid. कोणता amino acid कोड करतं? Methionine. Codes the amino acid methionine. If there will be the change, that is, if this AUG is changed GUA, या पद्धतीने जर ते चेंज झालं, or if we read the code, that is the GUA, okay, जर ते रीड केलं, that is it will be the different. So here GUA will be the different codon. GUA will be the different codon. So polarity means what? That is here the first codon, second codon and third codon, and these codons are always from 5' end to the 3' end. So here they having the polarity, that is the genetic code always in the direction, that is here 5' end to the 3' end. जर आपण 3' end to the 5' end रीड करत गेलो तर हाच AUG हा कोणता कोड अमायनो ऍसिड कोड करतो? मेथिओनाइन. जर आपण हा असा रीड केला GUA, that is it will be the wrong. It will be the wrong. Okay, so genetic code having polarity, that is they are read from 5' end to the 3' end. Then next characteristic, that is genetic code is non-overlapping. बघा आपण पहिलं ट्रिपलेट बघितला, दुसरा पोलॅरिटी बघितला, तिसरा आहे that is genetic code are non-overlapping. Non-overlapping. Genetic code are non-overlapping. So say that is here if we see, that is experimentally evidence who proved that is the genetic codon, these are the non-overlapping. So see that is how they are non-overlapping. So see here, that is here is the 5' end, here is the 3' end, that is of the mRNA. So here the first genetic code, that is for example here the first genetic code is the ACG. Okay, and second genetic code will be the here CGU. Okay, so here ACG, this genetic code, it mainly specify the single amino acid. Single codon specify single amino acid. Okay, so say that is here the ACG, it code that is the amino acid AA1, while CGU, this codon codes amino acid AA2. So it is the one amino acid, it is the second amino acid. Like this, one codon specify one amino acid. So here the experimentally evidence is that is for the overlapping codons. So here on the mRNA, if there are the 6 codons are present, first 3 codon, that is they code one amino acid, another 3 they code the another amino acid, like this. Okay, so these are the codons which are non-overlapping. Now overlapping condition मध्ये कसं असेल बघा, in the overlapping condition, that is here it is the 5' end, here is the 3' end. Okay, now overlapping condition मध्ये कसं असेल, so see that is here ACG. Okay, so here is the CG and U, CG and U. So here in the first codon, here like this, so it is the first codon which will be called the one amino acid. So overlapping म्हणजे कसं येईल, आता पुन्हा हा दुसरा कोडॉन तयार होईल CGC, हा दुसरा कोडॉन असेल. आता तिसरा कोडॉन कुठून तयार होईल, इथून G पासून GCG, हा तिसरा कोडॉन असेल. ठीक आहे, त्यानंतरचा कोडॉन कोणता येईल, इथून पुढे CGU. So it is the fourth codon. मग या पद्धतीने इथं जे काही कोडॉन्स तयार होतील, these codons, these are the, it is the overlapping condition. ही जी कंडिशन कशी आहे, it is overlapping condition. Overlapping. But genetic codon are non-overlapping. So this condition is non-overlapping. Okay, so here the genetic codon are always that is non-overlapping. ते non-overlapping मध्ये असं सलग 3 सक्सेसिव्ह न्यूक्लिओटाइड मिळून एक कोडॉन तयार होईल. त्यानंतरचे पुढचे 3 मिळून एक कोडॉन तयार होईल. यातला कुठलाही कोडॉन पुढे येणार नाही किंवा कुठलाही नायट्रोजन बेस हा पुढे जॉइन होणार नाही. दॅट इज दे आर नॉट इन ओव्हरलॅपिंग कंडिशन. दॅट इज दे आर नॉन ओव्हरलॅपिंग. जेनेटिक कोड आर नॉन ओव्हरलॅपिंग. कन्सेप्ट लक्षात येतोय. सो इट इज द थर्ड कॅरेक्टरिस्टिक दॅट इज द जेनेटिक कोड आर नॉन ओव्हरलॅपिंग. Then next characteristic that is the genetic code are always commaless. So see student, that is it is the fourth characteristic, that is genetic code are always commaless. Genetic code are always commaless. So commaless means without any gap. That is, there is no gap between the successive codons. No gap between successive codons. No gap between successive codons. So see, that is here how they are. So here that is the 5' end and here is the 3' end. Okay, so here the genetic code, that is they are, see here this codon again like this. So here A, C, G, U, G, C, C, G, U. So these are the genetic code. So here the 3 successive nitrogen bases, it is one codon. Then again the 3 successive nitrogen bases, it is another codon. So this codon specify one amino acid, this codon specify another amino acid. So here there are the 3 successive nitrogen bases. These are known as the codon, but in the codon there is no gap. There is no gap. So no gap between successive nucleotides. That is here the no gap. No gap between successive nucleotides. No gap between successive nucleotides. म्हणजे या न्यूक्लिओटाइड्स मध्ये गॅप नसतो. हे सलग न्यूक्लिओटाइड्स असतात. आणि लक्षात. सो फर्स्ट 3 that is one codon, then 3 another codon, then 3 another codon, like this. Okay, so the genetic codon, there is no any gap between the successive codons. So the genetic codes are commaless. Genetic code कसे आहेत? Commaless आहेत. Okay.